We will start with the introduction part of this study, the background of the symbiotic relationship between Gastridia elata and Almilaria gallica. Gastridia elata is a traditional and valuable Chinese medicine. It has anticonvulsant, hypnotic, antidepressant, and other pharmacological effects, and it has no roots, leaves, and chloroplasts, thus it cannot carry out any photosynthesis processes. It relies on the digestion of A. gallica invasion of its rhizoma for carbon and nutrients. For Almilaria gallica, it plays an important role in the carbon cycle and is beneficial for natural ecosystem as it acts as a saprophyte, which is the rotting fungi. And only a few of Almilaria species can be symbiotic with the G. elata, as the ability of different strains of the Almilaria to form rhizomorph and the ability of the rhizomorph to infest cortical tissue of G. elata differs. There are three objectives of this study. The first one is to sequence and assemble the whole genome of Almilaria gallica M3 strain, because the current study they focus more on the phenotyping, physiological and biochemical assay, and the transcriptome analysis. So the study wants to carry out the whole genome analysis of the genus Almilaria to understand more about the symbiotic relationship between these two plants. And the second one, to analyze the intrinsic genomic mechanism that enable A. gallica M3 strain to form a symbiotic relationship with G. elata. We want to study the gene family related to the host interaction. They are crucial in the growth and development of the plant pathogenic fungi. The third one is to provide a theoretical basis of the standardization of A. gallica M3 strain, because they want to provide the foundational genetic information that can be used to define and standardize this strain, which is important in supporting the growth and development of G. elata.
I will briefly present the materials submitted used in the genomic study of Almilaria gallica M3, a fungus strain provided by UNAS and how fungi industry in China. The strain was cultured on PDA solid medium at 28 degrees Celsius in the dark for 5 days and maintained at Kunming University of Science and Technology. Genomic DNA was extracted using the CTAP method, and its purity and concentration was confirmed using nanodrop and cubic fluorometer. Whole genome sequencing was done using Illumina, NovaSeq, and Oxford Nanopore methylene platform. Assembly was performed using ECAD, corrected using recombinant, and defined using PurgeHab lattice. Guppy software handled base calling, and sequences were filtered into FASIHAH format. Genome completeness was assessed using BASCO with the fungi ODB10 dataset with score above 90%. Repeat were identified using repeat masker and repeat modeler. tRNAs, rRNAs, and ncRNAs were annotated using tRNA scan SE, rRNA database, and Inferno respectively. Gene function were annotated using CAGE, GO, PFAM, and NR databases through Diamond Blast P and InterpoScan. Pathogenicity was predicted via the PHU database, and CZyme were identified using the KZ database. For evolutionary analysis, the fungus strain were compared using AutoFinder, MASL, and REXML to construct a phylogenetic tree. Gene family changes then were analyzed using CAFE. Lastly, comparative genomics between 3 A. gallica strain was done, and GO, CAGE enrichment was analyzed using cluster profiler in R. This method provided a robust foundation for understanding the genome structure, function, and evolution of Almilaria gallica M3.
Assalamualaikum. My name is SYAFIQAH binti IBNUL BAHAR with matric number A193293, and I'll present the first part of the results section of the article. The first part of the results explains the general genome characteristic resulted from the study. The genome size of A. gallica M3 is 861795663 base pairs, where 47.43% of it is the average GC content, and as much as 12,557 are identified CDSs. From the BUSCO assessment, they discovered that 95.4% genome completeness and 14 fragmented BUSCOs and 21 missing BUSCO. The predicted RNAs in the genome is 281 tRNA, 60 rRNA, and 25 snRNA. The genome sequence has been deposited in the gene bank with the accession number PRJNA 1082085. As for the gene annotations from GO, KEGG, and PFAM databases, GO analysis annotated 7,671 genes into over 18,000 terms, with the most abundant in the membrane component. KEGG mapped 2,557 genes, mostly in metabolic pathways. PFAM analysis annotated 7,308 genes, with the top protein families being major facilitator superfamily, cytochrome 450, and protein kinase domains. Next is the gene annotation using the NR, or non-redundant database. A total of 12,069 genes from A. gallica M3 were successfully annotated through this database. Most of them, about 62.68%, match with Armillaria gallica, which confirms the species identity of our sample. Other genes also showed similarity to other closely related species. About 18.65% match with Armillaria ostoyae and 16.38% with Armillaria solidipes. Only a small proportion match with other species. This kind of annotation is important because it helps us understand how closely the genome of our strain relates to other known fungi and supports the accuracy of our sequencing. Next is the C enzyme annotation in A. gallica M3. A total of 452 C enzymes were identified, mainly glucoside hydrolysis, which is 191, followed by auxiliary activities, which is 119, and glycosyl transferases, which is 73. This helps explain how the fungus breaks down and uses carbohydrates in its environment.
In this study, a total of 3,412 genes in Armillaria gallica M3 were analyzed to understand their roles in pathogen-host interactions using the PHI database. These genes were classified into 8 functional categories based on their predicted impact on the fungus' ability to cause disease. A maximum likelihood phylogenetic tree was constructed using RAML software. The analysis revealed that A. gallica M3 is genetically distinct from A. gallica AR212, but it shares a closer evolutionary relationship with A. gallica 012M. The evolutionary analysis of A. gallica M3 gene families was conducted using KFA. The analysis revealed contracted families included those involved in oxidoreductase activity, fungal type cell wall structure, monooxygenase activity, hydrolase activity, and the extracellular region. Families related to transmembrane transporter activity and iron-ion binding were significantly expanded. A functional analysis using gene ontology and KEGG annotations was performed. The shared genes in panel A are mainly involved in essential cellular functions such as carbohydrate metabolism, protein transport, and catalytic activity. The unique genes in A. gallica M3 in panel B are enriched in more specialized processes like iron-ion binding, fatty acid biosynthesis, and heme binding. In this study, we sequenced the 83.33 mbp genome of Armillaria gallica M3, composed of 38 contigs with an N50 of 6 mbp and a GC content of 47.43%. We identified 12,557 genes with repetitive sequences comprising 44.36% of the genome. Gene ontology analysis showed significant contraction in gene families related to monooxygenase activity, hydrolytic enzymes, and the extracellular region. These genes are often linked to pathogenicity, minimizing damage to Gastrodia elata inner tissues, and supporting a stable symbiosis. The genome contains 452 carbohydrate active enzymes, fewer than other Armillaria species, with enrichment in glycoside hydrolases and glycosyl transferases essential for nutrient breakdown.
Next, enzymes involved in plant material degradation and symbiosis. Glycosyl transferases and carbohydrate esterases play roles in glycosidic bond formation and modifying fungal chitin. Key enzyme families include GHs, CEs, and PLs. Genome analysis found over 3,000 PHI-associated genes, mostly linked to reduced or unaffected pathogenicity. This suggests that A. gallica has low pathogenicity, which favors its symbiotic relationship with G. elata.
So the conclusion and comments for this study, the first one is there are a total of 3,412 genes found that is involved in the host-pathogen interaction mechanism from the genome sequencing and assembly processes. The second one is from the study, they found out that the A. gallica M3 strain, it was weak to the pathogenicity and was suitable to mixing planting with the G. elata. The third one is A. gallica, it was composed of glycoside hydrolase and glycotransferase, which are helpful for the G. elata to obtain the potential of nutritional application. The fourth one is, in this study, only one A. gallica strain was sequenced, so further studies should be made with analyzing more strains from this species, as there is a potential that it could reveal the evolutionary trends in the symbiotic relationship.